All right, welcome everyone. Glad you could make it to this webinar for RDCA-DAP. We're specifically having this particular talk being on the value of integrated data in rare disease drug development. And we're going to focus on a case example in polycystic kidney disease. My name is Jackson Burton, and I'll be hosting for today. We will have a panel session discussion after the fact, but let me go into a couple housekeeping items just real quick. If any questions arise during the talk, please direct them into the Q&A section over in the chat panel. You'll see that there's a drop-down area for Q&A, and please make sure that you direct it to all panelists, just so we don't lose any questions in the process. So any questions that come up during the talk can be recorded there, and then any questions that we generate during the panel discussion, please direct them to there. Let's go get started. A little bit about myself. So I currently lead the quantitative medicine program here at Critical Path Institute. We oversee a number of different quantitative projects that relate to unmet needs in various indications, and our main drive is to generate quantitative solutions for these unmet needs using patient-level integrated data. The RDCA-DAP platform offers a great example of the intersection of our expertise with the platform, just to demonstrate its utility. So I'm going to be happy to go into that. Our speaker today is, on the next slide, Rhoda Mise. She is a stellar statistician and has been working with the Quant Med team for about 1.5 years. She's driving a number of different solutions across the board, and is a statistician by training, and has been driving this work here in polycystic kidney disease. So she's going to be stepping through the work that she's done with a little bit of background, as well as what motivated this work come time. So we're going to head and let me introduce the panel as well. So for the conclusion of the talk, we'll be having several folks for our panel discussion. We have Klaus Romero, who's our Chief Science Officer. He's been at CPATH for quite a number of years and brings a tremendous amount of experience. We have Megan Kalla, who is one of our pharmacometrician postdoctoral fellows, and has been doing some great work in the rare disease space. We have Jeff Barrett, who is our Senior Vice President, who is currently leading RDCA-DAP, overseeing all of the science operations and strategy for it. And we have Alexander Bertrand, who is our Scientific Director in RDCA-DAP, and who's going to be bringing his expertise in the rare disease space for the discussion. Okay, so with that, I'm going to pass it off to Rhoda. So Rhoda, please take it away, and we'll move on to the next slide.
Hello, everyone, and welcome to this session. So we are going to focus on the project within RDCA-DAP for polycystic kidney disease, and just a review of what goes on. So model-informed drug development serves as an approach to generate development tools, and it involves collaboration between 3 different partners. For example, the Data Collaboration Center within CPATH provides us with the data. The quantitative medicine team does the modeling, and our external collaborators within the Rare Diseases Forum help us and provide scientific knowledge and other knowledge on disease progression. So all of this working together enable us to develop the drug development tools. Next slide. So previous work, due to the integration of data within CPATH, has led to several regulatory successes. For example, using the integrated data, baseline total kidney volume was qualified as a prognostic enrichment markers for patients with ADPKD who were at high risk of renal decline. So the underlying evidence for the qualification of this baseline TKV was a joint model that comprised of a total kidney volume disease progression model and a survival model for 30% decline in eGFR. Next slide. Okay, so how do we leverage the underlying qualification evidence? So on this table, we have an example of how this is done. For example, for a patient aged 30 with a baseline TKV value of 1.7 liters, the table shows the time varying probability of not reaching a 30% decline in eGFR. And for a typical clinical trial of 3 years, for example, such a patient will have about 86% chance of not arriving at 30% decline from baseline. And the graph at the right-hand side just shows the plot of what we would expect. Next slide. Yes, so following this qualification, some conversation continued between CPAT and PKD consortium members, as well as the FDA agency, and this together with several clinical trials that use the evidence that was provided led to the designation of TKV as a likely surrogate based on the same evidence. So TKV was subsequently designated as a reasonably likely surrogate by the agency. And the title here is just reflective of what you will see on the FDA website. Next slide. So given this success, now we want to ask, how can we use TKV as a reasonably likely surrogate within a clinical trial design? Next slide. Next slide again. So to do this, we will need to quantify. So we will need to do some model that would link the biomarker within a disease progression model. And there is where the modeling work comes in. Next slide. Okay, so far we have shown that we were able to have regulatory successes based on the evidence that was provided prior. So such success has enabled sponsors to answer several important questions, which are highlighted in green, yes, but there are still equally important outstanding questions that have yet to be answered. So we still have these questions that are equally important. For example, if you look at the number 9, so far with the evidence, we are not yet able to use TKV as a reasonably likely surrogate. Next slide. So with this outstanding question, we ask ourselves, how can we address these remaining topics? So this is where CPAT within RDCA-DAP proposed to do this, to develop a clinical trial simulation tool with the following components: multivariate TKV and eGFR dynamics model, and this will be linked to a survival model for end-stage renal disease, ESRD. So such a tool will provide a means for FDA reviewers and sponsors to compare different clinical trial design considerations that are highlighted here, such as power and sample size calculation. Next slide. So with this goal in mind of developing such a model, we required a data set, and the in-house data set within CPAT that was used comprised of 5 observational studies. And if we focus to the table at the bottom, these 5 observation studies had 2,392 records for both the longitudinal markers, TKV and eGFR. These were from 587 patients. Out of this, 81 experienced end-stage renal diseases. Next slide. Okay, so the first step in our model building was to visualize the key variables of interest, the longitudinal markers, TKV and eGFR, and our end-stage renal disease time to that. So on the left-hand side, we see the visualization of the longitudinal markers, and at the top, we see that both eGFR and TKV on the log scale are symmetric and they are approximately normal. And as we see the plot of the longitudinal markers over time for eGFR, on average, we see that it declines, while the TKV on the log scale on average increases over time as expected from a clinical standpoint. On the very right-hand side, we plot the time to end-stage renal disease, stratified by the baseline chronic kidney stage. And also, as expected, those who are at a later stage or in the chronic kidney disease progress to end-stage renal disease earlier than those who are just at the beginning of this chronic kidney stage, earlier stages. Next slide. Okay, so the next thing was to build our model, and here we show equations. So it was a joint model comprising of the longitudinal markers, TKV and EGFR, and a survival model of end-stage renal disease. So while these equations seem complicated, they do have real-world meanings. Next slide. Yeah, so the first one quantifies the variability between patients, which is necessary for clinical trial design. The next one incorporates patient features for better predictive accuracy. Such features include baseline severity, sex of the patient, etc. This is important for informing the trial entry criteria. And the next is to describe the longitudinal measurements of TKV and EGFR rate of decline. These are important in informing the time to end-stage renal disease. And at the very end, we show the mechanism of how to predict end-stage renal disease. So this represents the trial endpoint. Next slide. Okay, so once we estimated the model, we, excuse me, we obtained the results. And as we can see here, these results have their estimates and their real-world interpretation. So if we direct our attention to the very bottom, next slide. Yes, so these are the time-varying effects of total kidney volume and EGFR rate of decline, which are our longitudinal markers. So for example, the TKV effect had a value of approximately 1.0352, which means at any given time, a unit increase in total kidney volume predicted value increased the risk of end-stage renal disease by approximately threefold. Next slide. So once we estimated the model, our next task was to evaluate the model, and we did this by the usual diagnostics and performance testing. So on the left-hand side, we have the plots of our longitudinal markers. So the diagnostics showed that the model performed well. For example, the residual plots, they are supposed to be symmetric around the mean, and we can see that was the case for both TKV and EGFR. And the predicted fitted TKV versus observed are supposed to be around the symmetric line, and most of the values are concentrated around the symmetric line. So the model appears to be properly describing the data based on these diagnostics. On the very right-hand side, we see the visual predictive plot on time to end-stage renal disease. And if we especially focus on the first 3 years, we see that our model is doing extremely well based on the observed and simulated the overlap. Next slide. So far, we have developed a model for disease progression, and our final objective is to build this into a clinical trial simulation tool. And this would incorporate the disease progression model that has been developed so far, which has things like the demographics and other values like EGFR and TKV. This will be linked together with trial components such as placebo, dropout, and trial design. And lastly, together with the drug effects, which could either be disease modifying or symptomatic, all this together will be built into a clinical trial simulation. Such a tool will be important to inform clinical trial design, where a sponsor could be able to choose the inclusion or exclusion criteria, several enrichment strategies, and trial duration and sample size. So just to show an example that this has been done within CPAC, on the right-hand side, we have such a tool which was developed within the Alzheimer's disease space. And we would envision such a tool for this particular project. Next slide. So in conclusion, we have shown that the quantitative tool fundamentally transforms data into knowledge. Within the context of PKD, RDCA-DAP has enabled the generation of such tools through provision and integrating of data, and these are important in addressing key drug development questions. So especially the collaborative framework within RDCA-DAP will provide a platform for open knowledge and data sharing, since it is a pre-competitive space. And with this, I will pass it over to Jackson.
All right. Thank you, Rhoda. That was a great presentation, and a lot of important pieces there that we want to touch on. So we have a good amount of time that I think will allow us to have some good conversation around this. We've had some questions come in, and I'd like to kind of kick things off with those. But let me first just remind you of the housekeeping endings again. For this discussion here with the panel, please direct your questions into the Q&A section, and please address it to all panelists, just so we don't miss anything coming through, and that way we can track those, pretty well. So I think we'll kick things off, right away here, and we'll go ahead and kind of show ourselves popping up here. Apologies for the, working remote has always led to some internet challenges. So if I suddenly am disconnected, I will still maintain my audio through the phone. So it's just the nature of working in virtual settings. So let's go ahead and get started here. I think the question that I think stands out as a good first one is really, what impact has this work had on PKD drug development? And I might direct that one at you, Klaus. I know you've seen a lot of evolution in this field over the past years. So let me start by maybe handing that one over to you, and we'll kick things off.
Thanks, Jackson, and thanks, Rhoda, for the insightful presentation. That's a pretty relevant question, right? So why are we doing this in the first place, and what kind of impact has this work had? As you know, there is a drug which represents the first drug to ever be approved by the FDA for slowing down the progression of PKD. That drug has been around for a few years. The work that Rhoda alluded to in the beginning that led to the qualification of total kidney volume as a prognostic biomarker for enrichment, and then subsequently the designation of total kidney volume as a reasonably likely surrogate, contributed to the meaningful evaluation of that drug. So that's a pretty powerful contribution to accelerating drug development for orphan and rare diseases. That's one. But then the consequence that it took once a regulatory decision was reached, that really piques the interest of investing by pharma into these conditions. And what we see now is a really robust pipeline for drugs under evaluation for PKD. That's fantastic. That's where we want to be. Now, recently there was a little bit of not so great news with one of those candidate drugs, but that's okay. And that highlights why we need to take the next step that Rhoda described, because the whole point is to ensure that just because you have a reasonably likely surrogate doesn't mean that all the problems have gone away for drug development in this condition. And what Rhoda showed is the evolution of the work so that sponsors can extract the most value out of that reasonably likely surrogate. And so the fact that we have a win and then another example of not so great news, it represents the call to action that this work needs to continue. So I'll stop with that. Back to you, Jackson.
Thanks, Klaus. Thanks. All right. Another question I have probably for you, Rhoda. I think there just wanted to be some clarification. Does the new model include EGFR as compared to the original work, which just had TKV?
Yes, that is correct. So the new model, if we can go to the slides, but the new model has both the longitudinal dynamics of EGFR and TKV jointly estimated with time to end-stage renal disease. So it extended on the initial work, which only had TKV. Yes.
Yes.
So if we go on the previous slide. Yes, so here we can see within the survival submodel, we incorporate both the total kidney volume and EGFR rate of decline. So there were two longitudinal markers linked together with time to end-stage renal disease.
Great. No, thanks for addressing that. Related to that, does the model, and maybe this is a question of the broader group, does the model that provided the evidence for the qualification and designation of TKV as a reasonably likely surrogate, originally include EGFR? And I think we've generally answered that, but just to be sure that the original qualification did or did not include that. I think probably a question for you, Klaus, on that front.
Sorry, can you repeat the question one more time, Jackson?
Just going back to the inclusion or non-inclusion of EGFR in the original designation.
Oh, right. Yeah. So in the original model, if you go back to, I think it was slide number 7, I want to say, where the decision from FDA is shown. One more. Go back one more. Yeah, this one. Right. So this was the first victory, really, and this represented the first win that represented really the power of being able to integrate the data, number one, and then leverage the data to transform the data into actionable knowledge for drug development, and the actionable knowledge is represented by the biomarker. At the time, the thinking was around the biomarker to inform TKV, to inform patient selection for clinical trials. So this model includes EGFR in 2 forms. One, as a baseline covariate that allows you to use TKV as a selection criterion together with baseline EGFR, and then a specific threshold EGFR loss at the end, 30% loss of EGFR over time, which was the binary endpoint for the joint model. What Rhoda indicated in the evolution of the model is that now there is a model that looks at the longitudinal dynamics of TKV and EGFR together, which allows you to derive any % loss of EGFR over time, and joined with a survival model for end-stage renal disease, which is a very clinically relevant endpoint later on in the course of the disease. I hope that answered the question, Jackson.
Yeah, that was great. Appreciate it, Klaus. A few more questions coming in. I'll direct this question to Jeff. So Jeff, how do you foresee this process speeding up drug discovery for complicated rare diseases where the biological mechanisms of the disease are not yet well understood?
I think what you saw in Rhoda's presentation is really, the accumulation of our best knowledge at the time into a useful tool. Now, the future for RDCA-DAP is to connect the platform with tools like this so that drug developers in the rare disease space can have access to them early on. So this will help with the early stage clinical development plans. It'll help in terms of finding the patients, understanding the diversity in patients, building detailed knowledge around inclusion-exclusion criteria. So this will really enhance the whole process. Plus, at the same time, our understanding of disease biology in the context of rare diseases. And again, it could be something where we borrow information from related rare diseases. So I think the benefit of having all of this kind of information, as well as the tools in an ecosystem that is collaborative and kind of spurs this sharing in a meaningful way, but also in the co-development of tools that enhance drug development. It will also be, I think, a benefit to early stage drug development from the standpoint of target identification, biomarker strategy, et cetera, because tools like this also showcase what the baseline is. So, you know, you may not be happy with where things are, but at least you have quantitative assessment of the current standard of care, and you have a target to shoot for in terms of improving things.
Thanks, Jeff.
Yeah, thank you, Jeff. I was going to say we have another question coming in, I think maybe for Rhoda. How does the method of TKV measurement affect the performance of the model? And is it valid only for ultrasounds or MRI? And the same question is also asked for EGFR, and they also asking which equation was used.
Yes, so I can answer for the TKV. So for TKV, we included the mode of measurement as a covariate within the longitudinal model. So it does account for whether it was measured by ultrasound, MRI, or CT scan. And then for the EGFR, I can't quite remember the equation which was used, but we did account for the race of the patient within the model.
Right, and I want to elaborate on that question because that's a really good one that speaks to the importance of harmonizing the data, because that the question is insightful. The volumetric estimations came from different techniques, and so as Rhoda indicated, it's not just a matter of taking the volume face value and running the model with it. You need to account for the actual way in which the volumetric measure was done as a potential source of variability, which was done. And then the EGFR metric, that was we had in some of the data sets an actual EGFR measurement in the patient level data, but we also had the creatinine level and you know, body surface area, BMI, and other relevant metrics in the data. And after discussing with the agency how to best approach the harmonization of the data, the actual EGFR was re-derived for all the data sets with the same method. And so that's what really provides with the reassurance of the data being relevant and sound and reliable in the background.
Thank you, Klaus. And we have a follow-up question. Does the blood pressure of the therapy feeds into the model as a major risk factor for further renal damage as a relevant in-exclusion criteria? So maybe just let me repeat that. Does also blood pressure and/or therapy feeds into the model, representing a major risk factor for further renal damage as a relevant in or exclusion criteria?
Yeah, that's it. Go ahead, Rhoda. You take that one.
Yes, so when we were harmonizing the data, blood pressure was not, how do I say, not available across all the studies that were there. So it was difficult to include since we had missing among our data sources. So it becomes, we did not, based on that, we were not able to accommodate it.
Yeah, and let me elaborate on that one because that's another really good question. What's the point of all this? To understand the behavior of the disease quantitatively, yes, but also to shrink the uncertainty. And that is represented by the residual variability in your estimations. And so you need to account for those sources of variability that allow you to control that, that residual variability. You will never, in this kind of an exercise, shrink that down to zero. And it's really important to be very transparent about the limitations that the data have. Blood pressure was one. So number one, it was not just what Rhoda indicated, which is an important factor, that there was a lot of missingness with the actual blood pressure values, but also the fact that in addition to the missingness of the information, there was quite a bit of uncertainty with the diagnosis of high blood pressure and the potential treatments, right? Because it's not the same to say somebody has normal blood pressure and does not have a diagnosis of high blood pressure versus somebody that has a diagnosis of blood pressure, is on anti-blood pressure medication, and is adequately controlled versus somebody that is not. And so that just was not adequately captured in the data, and that's feeds into the residual variability in the model.
Thanks, Klaus. We had another question come in, Jeff. I think this one might be directed at you. In order to speed up drug discovery and interest for ultra-rare diseases like ataxia, telangiectasia, not the correct pronunciation, what are the basic steps patient organizations need to do? Is it submitting patient data? What are the minimum requirements to benefit from CPAP tools?
So I think we have a mechanism to receive data. I mean, I'll actually queue up Alex to talk about our role in patient and group engagement, because soliciting this platform is only as good as the data they contribute to it. And you should know that the basis for the whole environment is the collection of diverse data sources. So that could be data from completed trials, data from patient registries, natural history data, and any number of data types. So a big part of the hard work under the hood is actually standardizing, curating that data, and making it available in a secure environment that protects patient privacy. So that's, again, a lot of the dirty work, but there is an ongoing effort to continue to get this data. And so one of the hopeful outcomes from webinars and other platforms such as this is to encourage sponsors, academic investigators to get in touch with us and to contribute data. A big part of the future success of this effort is to accumulate data. Alex, did you want to talk in more detail about the outreach effort?
Yeah, I mean, I think, as a rare disease platform, we want to be a place to help all the rare disease communities. So we're engaging in extensive effort to all communities and other also such as foundations, but also trying to create a network of collaboration with other data companies to try to aggregate as much data as possible for all rare disease. So this is the first step you can do to help understand our knowledge of rare disease progression is to share data with us. So that's part of the effort. I think second part of the question was what's the minimum requirement to benefit from CPAP tools. So there are two aspects to that. First, the RDCA-DAP platform is going to be open to the public in September. So we are launching a website, and scientists and communities will be able to create a login to the website and scroll through the summary of all the data that we have at the Rare Disease platform. And as a scientist, you'll also be able to request data sets and potentially have access to a workspace and do your analytical on the website. So this will be free of access, and that's a big benefit to rare disease communities. I think the second aspect to that is the work that we do internally, such as the tools that Rhoda presented today. And so these clinical trial simulation tools that we are developing are going to be available to the scientific community and clinicians for use for their clinical trials.
Awesome. Thanks, Alex. This might be a good segue for you, Megan. So I had another question. Are there other types of tools like this being developed on RDCA-DAP So maybe I'll pass it over to you, Megan, for the work you're doing in Friedreich's ataxia.
Great, yes. So one of the data sets that is hosted by the platform is the Friedreich's ataxia integrated clinical database. And that's one that I've been working closely with. So it's a compilation of various clinical trials and a very rich natural history study. And what we're trying to do with it is build a tool similar to what Rhoda presented, a disease progression model. And we're doing some endpoint exploration in this space as well. And a quick plug for what Alex mentioned, the showcase of this platform coming in September, where I'll be presenting that work on this pilot study in Friedreich's ataxia.
Thanks, Meg. And maybe just if you would add just a quick comment on at least the types of the discussions that it has enabled, and the importance of those discussions with key stakeholders, for really addressing kind of the endpoint conversation. I know this is sometimes a tough topic, but maybe just talk about the value of those kind of discussions and how it's really been directing the work.
Yes, sure. So it's opened up engagement with the FDA with regard to Friedreich's ataxia and the endpoints that are used in clinical trials. So there's some discussion involved or surrounding that. So what we're trying to do is to explore and engage with other key opinion leaders in this space to try and further to move the needle a little bit in helping to develop these tools.
Yeah, great. Thanks, Megan. Bettina, you had a follow-up question. I'll get it posted in a moment. And the question is it planned or for the trial simulation tool to include effects of relevant exclusion criteria like blood pressure or other CV risk factors, being aware of the difficulties of the interaction with treatment? That might be a question for you, Rhoda, but I could maybe at least kick that off a little bit. Based on the available data we have, we often will have challenges where if we don't have commonality of variables or heterogeneity of how variables were collected, it becomes hard to include those as informative predictors in these tools. But that being said, we do have some methods for accounting for data where we can deal with some missing data and still bring some additional added value. So in the specific context of blood pressure or other cardiovascular risk factors, Rhoda probably knows that data a little bit better and can speak to any potential of that or any challenges we may have, but that would probably be my very general answer. But Rhoda, if you want to maybe chime in there.
Yes, so since this is a limitation based on the available data, within the clinical trial simulation, we would have to include that that blood pressure was not considered within that. So that would have to be considered.
Yeah. So there's probably a good segue here because a lot of these questions coming up really do pertain to the specific intended application of these tools, and the types of these really good questions that have come up speak to the needs of sponsors, the needs of drug developers. And when we develop these tools, we develop them with kind of a regulatory pathway in mind, the goal of which is really to engage regulators, to engage industry, so that we have kind of consensus for a tool that could be fit for purpose in a particular context. And those tools really want to address the sponsor needs, but it's limited by the data. But nonetheless, once you have a robust integrated data set put together, you really can at least advance those conversations to develop these tools. Klaus, it might be a good segue here just to briefly touch on the regulatory mechanisms, maybe the concepts of a context of use definition, just to provide a little bit more background and context for this part of the discussion.
Yeah, happy to. And that's another great question from the audience. When you think about these kinds of solutions, you need to always think about providing the necessary scientific and regulatory confidence for these solutions to be adopted. You can work on many different solutions, but if those solutions don't get adopted, they're really not solutions. And so the question is, how do you generate that confidence on both the regulatory side and the industry side? For those of you that have an industry past like most of us, we as excited scientists can read the literature and can find solutions. They're published, and we would get all excited about, oh, this is the answer that I need to design my next clinical trial. I'm just going to apply it. Well, we all needed to have those conversations with upper management, and so at least in my experience, a good chunk of the time, and not all the time, the answer was not only no, but hell no. Because there's this perception that adding a new solution into the mix of a clinical development program may put the submission at risk, because there's uncertainty as to what the FDA or the EMA or PMDA might think about this novel solution, and the review time just may get extended until the agency gets comfortable with the science behind the solution that a scientist may get enamored with. Well, the whole point of these collaborative efforts is to provide that mechanism for connectivity with the regulators, to have these discussions about the proper intended application of these solutions. When you formalize that, you're going to hear the term context of use statements mentioned. That's really what it is. It's essentially the description of what the solution can be applied for and what the guardrails are. These solutions are not panaceas that can just solve any problem in drug development. They have very specific applications, and the definition of that application is what is so critical. And depending on the specific regulatory mechanism, the importance is to define that intended application upfront, learn from the data, refine that intended application, learn from the data again, refine the intended application again, until you reach a point where you have alignment with the regulatory agencies and with the sponsors on what the data can actually support in terms of generating a solution, and that's what you put out there. What you see on the slide is the final iteration of that journey for, in that case, the qualification decision for TKV as a prognostic marker. We had a similar pathway to get to the point of the designation of total kidney volume as a reasonable likely surrogate. And then now with this approach, the intention is to leverage a pathway that exists. It's called the Fit for Purpose Initiative, that is intended to review specifically quantitative drug development solutions. But in all cases, the intended application needed to be clear, and it needs to be based on what the data can support and what the data cannot support, and where the gaps are, and also a consideration for how the gaps can be filled, which is again an articulation of this continued learning confirmed paradigm. I'll stop with that.
Yeah, thanks, Klaus. It's really helpful to have that background context. I know CPAP has really done a lot in this space of advancing pre-competitive tools and quantitative solutions. So that's very helpful. I think we can maybe put out one last call for any questions, and we could move towards wrapping up. So I will continue towards the end. If we see any other questions come in, we will address them. But for now, I think we can jump to slide 21, which is the very end. And just a quick thanks to all for all those of you who asked questions and for our panel discussion. If there are any final thoughts from the panel members, invite you to just chime in here as we wrap up.
Just getting
all right
here. Just a final thought that this journey that you saw and the impact that has been achieved for PKD, as you see, started with one major win, led to another, and now we're after the next one. It's a journey, but you have to take the first step in the journey, and the first step starts, as Alex was mentioning and Jeff was mentioning as well, starts with the data. So if you definitely in the rare disease and orphan disease communities, you want to start the journey, let's have the conversation about the first step, which is the integration of the data.
Thanks, Klaus. All
Yeah,
right,
maybe
so
just
it's a good. Oh, go ahead, Jeff. Sorry.
Sorry, Jackson. Just a quick thought. I hope you've also seen the future from this platform, because I think having access to quantitative scientists such as Rhoda and the entire QuantMed team showcase what we're expecting in the future. And this is not something that CPATH will be doing by themselves. We have every expectation that we will partner with interested parties really in a global way to help this achieve its goal of accelerating rare disease cures. So I'm hopeful that you all will seek us out and help us build this.
Thanks, Jeff. On that note, may just conclude with an announcement on the next series within this particular series itself. This is going to be for the topic of how we can have RDCA-DAP inform optimal trial design, progressive rare disease. This will be a case study in Duchenne muscular dystrophy. Another member of the quantitative medicine team has been really driving this work, but not, as Jeff said, in isolation. Actually, several organizations have been involved in advancing this work and really showcases a lot of the collaborative potential that we can see in RDCA-DAP. So that'll be on July 21st, and we'll have Varun Agarwal, who's our Director of Statistical Mathematical Medicine, present. We'll also have Kevin Krudis from the Office of Clinical Pharmacology at FDA be on the panel session to provide some just thoughts from their perspective. So we'll be happy to have everyone join that for our next series. And with that, again, thank you all for joining. Thank you for the questions and the rich discussion, and we appreciate your participation. So with that, we will conclude here.
Everybody.